So for the students who are watching this, what is your name and position here at UT?
My name is Chris Yellman, and I'm an FRI instructor and also a lecturer at UT.
And what did you study for your bachelor's, master's, and PhD, and where?
I was a biology major at the University of Virginia, and then I took a big break after I graduated from college. I became a carpenter and started my own company and had a building company. I built and remodeled houses for about 10 or 12 years. And then during that time, towards the end of that time, I was thinking about how could I get out of this, because this is very hard work and I wanted something that was more intellectually challenging. So I thought about going back to biology. I thought about going back to architecture also. And I did a little bit of architectural drawing. But then I got back into biology and I started doing research as a volunteer in a lab. They paid me, actually. They hired me as a technician very quickly. And that kind of got me hooked. Because the project that I was working on moved along very quickly. It was very interesting. It took about 7 months and we had a publication. So I thought that was great. I thought that was normal. It's not at all normal to get a publication within 7 months, but I kind of got hooked on it. And then I went to graduate school.
What do you do in the Marcotte lab?
In the Marcotte lab, I'm a research scientist there. I officially, now I'm just a guest scientist there. But I'm a bench scientist, and Marcotte's lab is primarily computational. So I do experiments that complement the computational stuff that they do, where we test hypotheses that have been explored computationally, but where we would really like to get into the experimental side of things and test them. So genomics testing, mainly complementation of human genes in yeast. So the ability of yeast to live when human genes are transferred into them, the ability of those human genes to function.
So how recent is this research and how long have you been working on it?
This research got published about 2 years ago. It was about a 4-year project, so the work goes back 5 or 6 years. And my work that I contributed to this project was some genome engineering knowledge and experiences and tools that I had developed. And that goes back about 6 or 8 years to when I was a postdoc. I was a postdoc at Yale in Shirlene Roeder's lab. And Shirlene was a traditional cell biology genetics person, but she gave me enough freedom to do some technical stuff that I wanted to do. So I started developing some methods for doing genome engineering. Brought those with me to UT and started working with the Marcotte lab.
What is the broad application of the research that you are doing?
So there could be several applications. One, it's very nice basic science. It's very good basic science. There are some deep evolutionary questions about whether genes from different species that have diverged millions and hundreds of millions and even billions of years ago, whether those genes and the proteins they code for will still work when they're transposed from one species to another. That's a deep evolutionary question. And the question is, what are the constraints on evolution? How constrained is the evolution of proteins? After a billion years, will they still be doing the same function, and will they be transferable from one organism to another? So that's one of the basic science questions about evolution, and there are lots of kind of branches off that question about molecular evolution and how it happens. And then the other application potentially is that if we move human genes into yeast, they'll be making human proteins, that we can recreate human complexes or entire biosynthetic pathways in yeast that are done by human genes, or by genes from a plant, or by genes from a photosynthetic bacteria. And that then we could use those pathways to test drugs or to test function, and use them as kind of a model system, an artificial model system, a hybrid model system, but maybe a useful model system. We can test drugs on yeast that we could never give to people, for example. So it's possible we could create a model of a human genetic pathway in yeast, dump all kinds of drugs on it, and see how we can affect that pathway and fix diseases in that pathway.
Cool. Okay, and then what have been some of your results so far?
Well, so far we know that almost half of the human genes that we have moved from humans into yeast do function in yeast, and they will replace the function of the yeast gene. I have transferred most of the genes of the human proteasome. The proteasome is a big complex of about 32 different proteins whose function is to degrade proteins within the cell, usually regulated degradation, regulated by ubiquitin. Ubiquitinated proteins go to the proteasome to get degraded. And so we're building this complex. Most of the individual members of the complex in human will go into yeast and perform their function. And what I'm trying to do is move all the human components of the proteasome into yeast and have an entire human proteasome. And then when we have an entire complex, that maybe will be a genetic model system where we could test drugs or treatments or test the effects of human mutations on the function of the proteasome.
So how did you take all of this research and integrate it into the freshman research initiative?
Well, the projects are directly derived from my work. It's extremely direct. And what I have is when I get the students, 25 pairs of hands. They don't know anything about research in general, so I have to teach them all the basic stuff, and that takes a couple months maybe to even get started to be productive. But after that, I get 25 pairs of hands who can do a lot of the basic operations that I would normally do. Cloning genes, doing PCR, analyzing our strains to see if they have the genotype they want, whether we have modified the genome in the way we want. And also, I try to get those students to take up the intellectual aspect of the work, because they can search the internet just as easily as I can for publications that are relevant to the human disease effects or the biology that we're studying. So they may not understand the literature as well as I do, but they can go out there and search it and try to find interesting things to contribute to the lab.
Is there anything in particular that you think students benefit the most about from your lab?
I think what they benefit the most from being in a lab that's a unique experience that doesn't happen in chem lab, that doesn't happen in biochem lab, is independence. I think they benefit from thinking about the motivations of the experiment from the beginning, and to some degree, you're not completely independent. I wasn't independent as a grad student, even after a few years, you still depend on your advisor and others for feedback. But you become more and more independent. You can carry out the procedures more independently. You can think about the implications. You can troubleshoot a little bit. So I think that's the most valuable thing, is that this is it's like driving off-road versus driving on-road. Off-road, you have to solve all kinds of problems. You might run out of gas. You might get a flat. Your clone might fail, and you might have to help figure out why. So I think that's probably the most useful thing. Some people will be stimulated by that, and some people will find it very frustrating because there's a lot of failure in science, and you have to have the confidence to fail, solve the problem, and then succeed and move on to new questions. So that may be the most valuable part of the experience.
For those who aren't in FRI, are there other ways to get involved with your lab or research?
Sure, you can come as a guest in the summer. You can get involved in research by talking to any professor that you want to. There's nothing to stop you from walking in and knocking on someone's door. Introduce yourself, know something about the work they do before you go, and say, will you take me on? I can do this, this, and this, and most importantly, they want to know, will you be reliable? If you'll be reliable, they'll get you started doing simple things, and if you do those, you'll do more complex things, and you'll just progress as quickly as your level of progress and desire lets you progress.
What made you decide or become inspired to become a research professor?
It's the fact that I don't want to be a lecture professor. You keep up with things, you learn a lot when you're a lecture professor, but you don't get to practice it, and I am a hands-on kind of person. So I would much prefer to be doing things than teaching about them, although the teaching is a great way to learn. You fill in gaps in your knowledge, so there's nothing wrong with doing a little teaching, and I will do a little bit of that, but the fact that I like to do the hands-on work, and I like to be discovering things that people haven't done before, trying things people haven't done before, the novelty, the discovery, the challenge. I like the challenge of bench work, making it work when it's difficult, and I guess that's the reason. And then, FRI, I have this potential resource of all these students who can do work for me. So It's a bit like herding cats at first, but if you can herd cats, they can be quite productive. I don't want to herd sheep because sheep don't think.
What were some classes you took as an undergrad that you would say have helped you in your profession right now?
What are the most useful things? I should have worked in a lab. I didn't. That would have been very useful. That's something I recommend trying. Work in a lab. And hands-on. Do whatever you're interested in. Try doing it hands-on. Go with the people who are actually practitioners and find out what they do every day.
Can you give a few examples how any of those classes are applicable today?
Well, there's a bit of a reasoning process when you get to solving problems using genetics or even cell biology problems where it's observing cells and testing. You have to have quite a rigorous thought process about establishing a hypothesis and how you're going to test it and what the proper controls are. It's a little bit like a game of chess even. You can make moves and then you have to consider the implications. Did you properly do the controls to interpret that move? Because if you didn't, you're not really doing an experiment. So thinking through the implications and possibilities of your experiment, it's a bit like math or chess or strategy. People will do things differently. Some people will set up different kinds of controls. They'll be interested in different questions. I would say I learned the most from my graduate advisor, Dan Burke. He was a very good designer of experiments, great imagination for designing experiments, and he would find an interesting question. There would always be an interesting question, and then you would try to devise a method to trap that question and confine it to a certain experimental space that you could test. Questions are like, they escape. Hypotheses escape if you don't have the net there to keep them in, and the net is all your controls and your thinking and your careful experimentation. And he was very good at that, and he was very good at identifying key questions that could be done in a very direct and interesting way, and I learned a lot from that. That was very important.
What academic and or work ethic advice would you give current undergraduates based off of your own experiences?
Well, I have a list of personal qualities for research success, and I can't remember them all right now, but first of all, I think you need to be brutally honest. Not everybody in science is by a long shot, and it's very disappointing. But if you can be brutally honest with yourself about the results of your experiments, if it didn't work, it didn't work. Don't trick yourself. Because the faster you can admit that something didn't work, the faster you can move on to the next question, which might work. Or what didn't work might push you in the direction of what does work. So being completely honest with yourself about the results, the experiments you're doing. Being a finisher, don't leave something unfinished, because that last part of the experiment might be the part where you close the loop and you actually realize something. Instead, if you do 90% of it, you may never be able to reach that conclusive point where you can draw a lesson from the experiment. So you may think an experiment failed, but follow it out as far as you need to reach a conclusion. Spend some time reading. You can spend 2 months at the bench finding out something that you could have read in a journal and learned in 2 hours that somebody's already tried it, or they already know the answer, or there's some reason that your experiment won't work. So think your things through. Don't be afraid to take the first step. It's like anything. The first step is a motivational step. You say, I'm just going to go ahead and do this. And once you do that, you're involved in it, and the momentum of doing it, it gets interesting. But don't be afraid to venture into the unknown. Try new things. Constantly read new things and try new things in science. Not completely new. There are people who think, novelty is the thing, so I'll do something completely. You may not learn anything from that, but branch out within the distance that you'll learn something from it, and challenge yourself. Like, we're not going to start working with human cells in this lab. that would just be too far for us right now. We would sacrifice all the stuff we can do. It would be interesting, but we probably wouldn't get much out of it.
Personally, for you, what were some of your undergraduate struggles, if you had any, and how did you overcome?
I definitely had some undergraduate struggles. My biggest one was figuring out what I really wanted to concentrate on. And that's part of the reason I graduated from college and didn't pursue an academic job. You know, because I was interested in, I actually almost majored in government and history. And it took some foreign languages, I took my math, science, some literature. I had stuff all over the map that I was interested in. Architecture I was interested in, never took any courses. But just focusing on an interest, it took me till I was in my mid-20s or late 20s to think, okay, there are two things I'm really interested in, architecture and science. And even then I hadn't decided between the two until I started doing research. So that was the biggest challenge for me.
What are some of your future goals or aspirations outside of your research?
Outside research? I got some work to do on my house. Students are coming over to my house at the end of the semester. I have numerous unfinished projects, like a garage that I built this summer. I still have to do a little bit of work on it. I have a patio of stones that I'm working on. Outside of the lab, just to keep my kids happy. Make sure that they have challenging stuff to do.
Is it hard to balance your research and life?
Not too hard, not too bad. They like what I do. Sometimes they'll even come in here. So if you have trouble balancing your kids and your work, get them more involved with each other. You know, get your kids. You can get your kids involved with your work. You don't want to push them into it, but let them do the fun stuff, and that allows you to get a few things done when you're with your kids. Kids actually like that kind of thing. They like coming in here and looking on the microscopes and seeing stuff. So then I can be here in the lab working while they're messing around. I guess it's not too hard for me to balance it.
Since you're so far along in your career, what is the next step you expect to take?
Next step this year is to definitely publish at least 2 papers. And I have a backlog of work that I've done that's all publishable. I should be able to publish 3 or 4 papers in the next 2 years, and I have to absolutely focus on that. If I can do that, then the stuff that we're doing now with the FRA students this year will be the next round, and we'll publish that. It's quite a treadmill. You have to think a couple years ahead. It takes a couple years to finish a project, sometimes as long as a year to write it up and get it published. So the next step is to start the treadmill of publishing the stuff that we've done here and the stuff that I've done the last 4 years or so. It's been my independent work. I do some collaborations, so I publish with collaborators, but I haven't published very many first author papers here. So that's the thing, get them done. See if see if we can get funding for this lab would be another big thing. See if I could get outside funding, outside of the university, from a disease organization like the American Porphyria Association or the the American Cancer Society. They give grants. The National Science Foundation gives grants. If we could get something that would bring us in just 20 or 30 or 40,000 a year, would make a huge difference to what we could do here.
Has working with students changed your outlook on life?
Not so much. It hasn't really changed it so much, but it's made me aware of their outlook on life, which you don't know until you have contact. And their outlook on life is probably quite a bit more focused, most of them, than I was as an undergrad. And it's probably changed a little over the years that students are a bit more encouraged to choose their career path early and stick to it. For some people that pays off, for others it might not. So their perspective on life, I don't know, it seems like they have a pretty good perspective on life, most of the students. Maybe I had a good perspective on life, it's hard to remember.
What kind of technical problems do you encounter in this lab, and how do you overcome these problems?
So a specific scientific challenge. So when we modify genomes, the organism responds to that. And that creates complexity that you don't anticipate. So for example, we can move human proteasome genes into yeast, and the yeast appear to be functioning normally, they're actually not quite normal. And if they're not quite normal, then those strains can start to compensate by creating mutations or by selecting mutations or selecting extra chromosomes that give them additional proteins to use. So one of the technical problems with this is making sure that the strains we make have the genotypes they have, that we expect them to have. Human gene in the genome and no other changes in the genome. We'd like to have that. But there are many, many reasons that push cells to make other changes in their genome when they're confronted with some kind of selection pressure. So that's one challenge. Constantly solving small challenges in cloning and DNA assembly, how to put things together correctly. I'd say those are small challenges. Thinking in a really big picture sense, that might be the biggest challenge. You know, how important is this work? How good is it? Will it lead to enough interesting things that we should really devote ourselves to it? We have to make a few decisions sometimes, like which organisms to work with. You know, this could end up being really interesting. It's a prokaryote, but it's photosynthetic. Can we work with it and discover something? Maybe. So,
how long does it take to get students ready for advanced research?
advanced research, advanced is a relative term, but I would say my students who've been with me a year now, who are mentors, are very productive, potentially, if I can make sure that I don't set them up with something that has a lot of unknowns that are going to really create trouble. But they can get enough experience in a year to start doing quite productive work for me. And along the way, even in the first semester, I can get some productive work out a little bit. But it's not advanced research. We have to be careful. The only time this will become advanced research is if we can put it all together and it's really a meaningful piece of work. Because we could do all these things, we could do all these procedures, and if we don't think about strategy and the big picture, we might never integrate this into a meaningful piece of research. So that's my job, to try to make sure, collect all the work we're doing and make sure it's always focused toward the meaningful questions. So I'd say students and the administration should be honest with you about what you can do. I mean, let me give you an example. Just working on a construction site, your first year as a carpenter's helper, you're carrying wood, driving nails, and stuff like that. You are not laying out the building. You are not making decisions about where materials should be stored or how to set up a particular part of the job site. And that's carpentry. It's not as complicated as research. So it's a long process. And it's just, you just advance.
Since your research focuses on something so specific, and most students don't cover these in general science classes, how do you ensure that your students follow the concepts that are covered in your lab?
So how to give them a good education that's more general. So I try that by my little book. I'll show you my little book this time. It's our little book about your personal habits for success in a research lab. Common sense, honesty, don't make assumptions, always do your best, have clear goals, blah, blah, blah. Then I have a list of basic molecular biology skills. The reason I have this is a list of things that you might teach in a typical lab, in a more traditional lab. They might teach you all these things. We do them in here too, but we don't do them in a predictable order. We don't always do them very thoroughly, and you may not cover all these. But if you look at this list, any student can say to themselves, well, I haven't done a plasmid prep yet. I should learn how to do that. That's on the list of basic skills that I should know. Do I know my sterile technique? Do I know how to prepare solutions, basic solutions, pHing and concentrations? Do I know how to do PCR, DNA purification from genomic sources? If you do that list, you've gotten a pretty good education in molecular biology. That will cover you. So there is definitely a conflict in FRI. We're trying to do original research, but we want to teach you too. And teaching is a different thing. Teaching is learning fundamentals. So you have to try to find out where are the fundamentals in the things we're doing. So DNA purification, PCR, ethanol purification, making oligos double-stranded, annealing things, melting things, multi-piece assemblies. Those are all basic techniques, so you can get that education if you pay attention to it. And if you're not doing it in your project, go tag along with somebody who is doing it. If they're doing genomic prep from this blue-green algae, tag along with them and see how it works. And then you'll have gotten a bit of an education in it.
Well, I think that's all we have, so
Yeah.
you did great.
Okay.